Let's talk lungs with Trent and Lori from Lung Saskatchewan. Dedicated to connecting you with lung health experts and key opinion leaders who will share current evidence-based lung health information to inspire you and those you care for to take your best breath. This podcast is recorded on Treaty 6 territory, the ancestral and traditional territory of the Cree, Dene, Blackfoot, Saulteaux, Nakota Sioux, as well as the territory of the Métis.
On this episode of Let's Talk Lungs, we will be speaking with Dr. Kyle Brown. Kyle is the Associate Director and Field Medical Advisor for Moderna Canada and brings his expertise in medical affairs and medical information to our conversation today. Kyle obtained his B.Sc and Ph.D. in Medical Microbiology from the University of Manitoba, as well as completed a post-doc research role with the Vaccine and Infectious Disease Organization at the University of Saskatchewan. He has a broad history of working in the pharmaceutical industry with both U.S. and Canadian-based healthcare providers. Kyle's experiences focus on infectious disease, human and animal vaccine development, molecular biology, mRNA technology, and cardiovascular-based pharmaceuticals.
This episode of Let's Talk Lungs is sponsored by Moderna Canada. Moderna's mission is to deliver on the promise of mRNA science to create a new generation of transformative medicines for patients. Thanks for joining us for today's episode, Kyle. Since the COVID-19 pandemic, we have been hearing and learning more and more about mRNA technology. We know that Moderna has been in the forefront of mRNA, and we're excited to talk more with you today. Before we get into the science and the therapies, though, we do want to know more about you. Tell us about your education and your interests, and what led you down the path to what you're doing and leading on today.
Thanks, Lori. So I was born and raised in Manitoba. So my post-secondary education included an undergraduate degree in microbiology as well as a PhD in medical microbiology at the University of Manitoba. And while I was doing my PhD, I had the fantastic opportunity to work at the National Microbiology Lab here in Winnipeg with high containment level viruses that cause hemorrhagic fever. And more specifically, I worked on the development of both molecular tools and a candidate vaccine for hantavirus, which is a virus that some may have heard. It's carried by rodents and makes a lot of people very sick. And so did that, and then I did a short stint working on animal vaccines at the Vaccine Infectious Disease Organization, or VIDO, at the University of Saskatchewan. And while I really enjoyed working in a lab on the initial kind of early discovery phase of vaccines for the better part of a decade, I really wanted to, have the opportunity to do something different and work more with products that more immediately made an impact onto the health of patients. And so I found the opportunity to work in the pharmaceutical industry within medical affairs. And so I made the jump over there, first as a medical science liaison and then working with medical information. And I spent over 7 years in the cardiovascular medicine space. And I had the unique opportunity to learn kind of about the U.S. healthcare system, travel through the U.S., and speak with a lot of high-level interventional cardiologists, and enjoyed it very much. But of course, then the pandemic hit and everything really changed, and it made cross-border travel challenging. And so an opportunity came up actually to join Moderna and actually work with, at the time, their recently approved mRNA-based COVID-19 vaccine. And so I jumped on it because it really brought me back to the vaccine space, which I found I'd kind of been missing a little bit. And so it's actually kind of funny because when I started working on my PhD at the NML, they had actually just finished the outbreak response to the original SARS coronavirus outbreak back in 2003. So now that I'm working on prevention of SARS coronavirus 2, it kind of feels like I've gone full circle back to where I began with my studies. And so since joining, Moderna over 3 years ago now, I've had the opportunity to really see what mRNA can do, and I've been excited to see the impact that we had for the health of Canadians and the future opportunity to kind of be part of the mRNA revolution in future potential medicines. And so, but when I'm not working, with healthcare providers, when I have some little free time here and there, I do enjoy traveling. I like camping out in the wilderness. I like to spend a lot of times with my 3 kids. They keep me busy most of the time. And so with them, it's a lot of reading and sports, watching movies, doing whatever I can to keep the time busy.
Sounds wonderful, actually. I want to back you up. Let's start, Kyle, at the very beginning of all of this. Tell us exactly what we are talking about when we hear mRNA. Can you give us a bit of a background on the mRNA technology? Because I'm not sure certain people realize how long it has truly been around.
For sure. So mRNA stands for messenger RNA. And so it's a natural molecule that exists in all of our cells. And actually, at any given time, we have millions to billions of mRNAs being expressed in our body. But, in order to understand the role of mRNA, we kind of need to take a step back and talk about how our bodies actually work. And so every function that our bodies take out, requires protein. And protein are used for the structure of our body. It's used for the function of our different organs, our heart, our lungs, our brain. It's used for the enzymes that digest our food, for turning molecules that we don't need into something that we do need. And it's also responsible for how our immune system works. So without proteins, we just wouldn't be able to exist. And so the question comes down to, how do we actually create the different proteins that we require? And so if you indulge me in a moment, just kind of an analogy, you can kind of think of the cells in our body as being like little factories. And so each of these factories has a head office where the blueprints are securely stored. And these blueprints contain the information for any type of protein that we could ever need. And so these blueprints are actually our DNA. And where they're stored is the nucleus of the cell. And so when we need a protein to be created in our cells, we don't actually want to take the DNA from the nucleus and move it out of there, because if it gets damaged or something happens to it, we lose that original information. And so instead of taking it out, and that's actually what happens in the case with cancers or things that damage our DNA, we no longer have the ability to create the proteins correctly. And so then the cells are a lot of times destroyed. And so rather than actually removing the DNA from the nucleus, we want to send out a message. And so they take a copy of the instructions from the DNA, they go out of the nucleus into the cytoplasm where proteins are made. That message is taken to the machines that make proteins, which are called ribosomes. And then the ribosomes create the protein that's needed. And when the messenger is no longer required, then that's actually destroyed. So that's what mRNA is. It's that temporary message that communicates the data from our DNA to the machines that actually make our proteins. And so although mRNA has been around for a long time, for as long as we know, because it's required for every living function, we only really discovered it in its function back in 1961, which is almost 65 years ago. And since then, we've actually been researching it, how it can be used as a therapeutic. And actually, the first injection of mRNA into animals as a therapeutic occurred back in the 1990s. And so, but the problem with mRNA or RNA as a therapy is that unmodified mRNA actually creates really strong immune response. And so essentially you put it into our bodies and our bodies destroy it before it can really do anything. And so one of the big advances that have kind of made mRNA technology into what it is happened back in 2005, where there was two researchers, Drs. Catalin Carrico and Drew Weissman, that discovered by chemically modifying the mRNA, they could actually prevent the immune system from destroying it. And so this is actually what made mRNA safe and stable enough for use in vaccines. And they were actually awarded the Nobel Prize back in 2023. And so that discovery, along with the simultaneous development of lipid nanoparticles, which is kind of the fat bubble that encapsulates and protects the mRNA and allows it to enter into our cells, allowed for the first mRNA vaccines for COVID-19 to be approved back in 2020. So overall, the pandemic really accelerated the awareness of the technology because a lot of people hadn't heard about it at that point, but really the technology itself has been under development for decades.
Let's talk a little bit about that term vaccines. It's been water cooler talk, obviously, since the pandemic, and I think one of the most searched words on Google in the past few years. So can you just give us a quick one-on-one on how vaccines work, and then how do mRNA vaccines work?
Sure. So vaccines essentially serve as a role of kind of priming or training our immune system. And so by providing your body with something that's similar to the real thing but won't make you sick, your body's more prepared to quickly eliminate the actual virus when you're exposed to it. And I say virus here, it could be a bacteria as well, but for the sake of simplicity, I'll just say viruses. But so essentially you get the protection from this vaccine, and the vaccines very rarely prevent the infection from occurring, but they do allow allow your body to clear the pathogen before you get severely ill and have complications that require hospitalization, that could have long-term complications, things like long COVID, or even potentially causing death. And so traditional vaccines have been kind of a couple different formats. There's been vaccines that have used weakened or inactivated viruses. And so this is the case with influenza vaccines. And so in this case, the viruses are killed or they're modified so that they can't cause disease. There's also protein subunit vaccines. These are, instead of exposing the immune system to the full virus, you actually get exposure to just the small component that you want the immune system targeted towards. So it's typically a protein on the surface of the virus that the immune system first sees that you want to target. And there's some limitations to this. If you have a weakened virus, if you put it into an immunocompromised patient, it can actually reactivate and still cause disease. They can't actually clear it. Inactivated viruses, when you kill a virus, it no longer really identically looks like the original living virus. And so it may actually not create as a specific immune response as you want. And protein subunit vaccines, you need to create huge amounts of protein outside of your cells. So usually they use animal cell lines or other types of ways to produce these proteins. But no matter how the vaccine is created, the whole point, again, is that it recognizes this protein as being foreign. Your body mounts an immune response, and it remembers it for the future so that when you are exposed to it again in the future, it's able to create the antibodies and the different parts of your immune system that are required to quickly eliminate the infection. And so when it comes to mRNA vaccines specifically, we all know during the pandemic when we've had our COVID vaccines, you go and you get an injection in the arm. So the vaccine is basically, again, those lipid nanoparticles, that kind of fatty bubble that contains the mRNA inside, and that's injected into your muscle. In the case of the SARS coronavirus 2 vaccine, it's the spike protein from the virus. And so those go into the cytoplasm of your cell. Again, back what I started with the beginning, the machines that make protein in your cells. And so it gives the message to the machines, it creates that protein. And then once the protein is created, it actually is then exposed on the surface of the cells. And so then the circulating immune cells can actually recognize it and create a response from it. Or it could actually be excreted from the cell out into the circulation, where then it moves around and is again picked up by the immune cells. So therefore, instead of injecting ourselves with viruses or proteins that we want to create the immune response against, we're actually just injected with the message of how to create that protein using our own machinery within our bodies to create those proteins. And so that's kind of the big difference between a conventional vaccine and an mRNA vaccine. You're actually just providing the message of how to create it rather than actually providing the virus or the protein itself.
Well, Kyle, we're digging in. And now our next question is, what is it about mRNA technology that does make it such a good fit for vaccine and other therapies?
For sure. mRNA has the attributes of a lot of kind of conventional therapies in the fact that you do get a very predictable dose response effect. So if you provide more of it, you get a stronger effect. You can compare it, for example, to Tylenol. When you take a drug like that, you take a higher dose, you're going to get a stronger effect. You take a lower dose, you're going to get a lower effect. It has a limited duration of effect, so it only lasts for a certain period of time. And you can redose it. So, when the effects wear off, then you can actually get an additional dose and get the response back. And so because of that, it can be used in many different ways, both as a vaccine as well as kind of different therapeutics. One of the big advantages of the technology when it comes to vaccines specifically is that mRNA can be tailored very quickly and accurately to target a specific disease or a new variant of a virus that's circulating. So one of the first things that happens when you identify a new virus is that virus is sequenced. And so you have the genetic code available. And so with that, development can begin within a few days. You don't have to waste time on trying to figure out how can you create the protein, how can you create large amounts of stable protein. How can you actually amplify and create large amounts of the virus? You can start with that initial stage and work very quickly. And that's ideal, especially during the pandemic. It was essentially about an 11-month period between when the vaccine was actually created, clinical trials were run, and the products were approved. And so that speed is a very important component of mRNA. The other thing is really around safety. And so, there's no need to handle live virus or use animal cell lines. Really lowers the risk of contamination in the final product because the mRNA vaccines really just contain things that your body already has. So salts and fats and sugars and the nucleic acids that make up the mRNA. And so it's very, a clean, relatively speaking, product that can be injected. Scalability is important too because, as I said already, you're using your body's own machinery to create the protein of interest. So you don't need a very large amount of starting product. Essentially, your cells themselves kind of serve as the amplifying step. And so this means that you can produce very small batches of mRNA that actually may be as small as targeting a single individual, but you can also scale it up to millions and millions of doses like we required in the pandemic. And so that scalability uses the exact same ingredients, the same equipment, everything, to create either that very small amount or that very large amount. And so the scalability is fantastic. You don't need all of a sudden a completely new factory or bigger bioreactors or things to get larger doses. You can create what you want on the scale that you require. And then finally, the versatility, I said already, there's so many things that you can do with it. You can use it to create simple proteins. You can create complex proteins. So there are some therapies where you actually have multiple mRNAs that encode for different proteins, that when all those proteins are created, they come together and actually form like a big complex. You can actually target, as we've talked about, external foreign proteins like from viruses to act as a vaccine. But you can also target internal proteins, for example, that are expressed in cancer cells. And then rare disease is also kind of another way that you can kind of replace missing enzymes or proteins that are required for that patient to have a normal metabolism as well.
Well, we are going to take a short break and get right back to our conversation with Dr. Kyle Brown.
Lung Saskatchewan has long been associated with leadership in health professional continuing education. Once known as a conference, Inspired Breathing now represents Lung Saskatchewan's renewed commitment to providing continuing education events for healthcare professionals that are responsive, innovative, and centered around partnership. Inspired Breathing inspires innovation, alignment, and partnership in lung health professional education, leading in transforming care. Find out more at lungsask.ca.
Well, welcome back, listeners. We are having a great conversation with Dr. Kyle Brown and learning a lot about mRNA technology. So, Kyle, we just talked about why mRNA technology is good science for vaccine and other therapies, and you talked earlier about the relationship between mRNA and DNA. Do mRNA vaccines affect the person's DNA?
So thank you for bringing up that point, that about mRNA vaccines affecting a person's DNA. And really because it's one of the main pieces of misinformation that really circulates online. And it frequently leads to patients being vaccine hesitant, especially when it comes to mRNA vaccines. And so as we talked about at the beginning, mRNA never enters the nucleus of the cell where the DNA is stored. It stays in the cytoplasm, and then it also is degraded fairly quickly within hours or days after it enters into the cell. And so the mRNA does not integrate with or alter the DNA in any way. It's kind of a one-way street from the DNA to the mRNA leaving, and then the proteins that are being created. And so the misconception may arise from a few things. There's talk about gene therapy. And so in gene therapy, the point of that is to actually make permanent changes to the DNA. So a lot of times that's a problem right from birth. They maybe have the wrong sequence in their DNA for a particular protein, so they can't create that protein correctly. And so the goal of a gene therapy would go into replace that part of their DNA with an actual functional copy of the information. And so mRNA vaccines are not gene therapy. They don't work that way. They stay within the cytoplasm. The other thing is there's certain viruses, more specifically HIVs, that are the biggest one that actually is known to take its RNA and to integrate into our DNA. And the reason it's able to do that is it actually brings its own enzyme with it that allows that process to occur. And that enzyme, called reverse transcriptase, is actually one of the major targets of antiretroviral therapy. So to prevent the incorporation into our DNA, they prevent that from functioning. And so we know that there are multiple independent studies, and the regulators, so Health Canada and this country, confirm that mRNA vaccines are safe in this regard, that they do not affect a person's DNA. But the important thing is there have been a number of surveys done that really show that people who believe that mRNA vaccines affect DNA have a low overall confidence in the safety of mRNA vaccines and kind of a lack of understanding of how they work. And so conversations with these individuals, you really shouldn't just disregard, their belief. You should really focus on how mRNA vaccines work. And I know at Moderna, we're working on creating a number of resources to make it easier for healthcare providers to be able to explain to their patients the basics of like the things we're going over here today. And there should also be a conversation about the wealth of safety data that has been generated, since 2020 when these vaccines were first introduced. And despite this being a relative newer technology, especially compared to the conventional vaccines on the market, billions of doses of mRNA-based COVID-19 vaccines have been administered globally. And so this isn't like a normal thing where you would take years and years to generate this kind of data about the safety of the product. We actually, in a very short period of time, had many, many doses being administered. And so that's given us a very good understanding about the overall safety of this technology.
So we're clearly hearing that mRNA vaccines do not affect a person's DNA. Does an mRNA vaccine have a side effect?
Yeah, so like all vaccines, mRNA vaccines can cause side effects, most of which are short-lived. And so these side effects are kind of what you typically feel after any vaccine. You kind of feel like the chills potentially and fever. You may feel aches and pains. It's why a lot of people, when they get a vaccine, they kind of think that maybe they got the actual virus itself because they feel like they're sick. But really, these are just signs that the immune system is working as expected. And the reason we feel like that when we are sick is because the immune system is going into overdrive and it's making us feel like that. And so when we get the vaccines, we essentially have a mini version of, how our immune system is reacting. And so that lasts for a couple of days. And some people, it's more severe than others, but it's nothing that is very, a serious complication. And so there are some serious side effects that are very rare because as I said, we've had billions and billions of doses administered. We kind of have a really good idea of the frequency of some of these very rare side effects. And so, one of the examples of that was with myocarditis and pericarditis, which was inflammation within the heart. And so back when the vaccines were first administered, we saw an increase in this in certain populations. And it was originally attributed to the mRNA vaccines, where as we gained more and more information, we learned that actually these side effects are actually also caused by infection with the virus too, and actually at a much higher level than what you see with the vaccines. In addition, there's actually the same side effects with the non-mRNA COVID-19 vaccines. The protein subunit vaccines have the same side effects. So a side effect like that could be potentially more due to the virus, the spike protein that's actually being encoded as opposed to the technology itself. And this is something that as we get more mRNA vaccines being created, more mRNA therapies being created, we'll get a better idea of which of these side effects is attributed to the actual technology and what is actually attributed to the target of that particular therapy.
Kyle, thank you for providing so much more clarity on what an mRNA vaccine is. Let's now talk about the advantages in using mRNA technology for the development of vaccines as well as other therapies.
For sure. So I think I touched on a lot of the points, kind of in an earlier question, just about some of the advantage of it. But I'll, kind of highlight again, kind of those key points is really the fast response. And I'll use kind of a practical example of the influenza vaccine. So currently with the influenza vaccine, they need to make decisions about the variants that are targeted a long time in advance to when the vaccine is actually used. And so sometimes they're looking at the viruses that were circulating in the Southern Hemisphere. They're looking at the previous year to try to determine. And so when the vaccines are actually available, they may no longer accurately reflect which variants are actually circulating. And so with mRNA, we can actually have a vaccine created and within arms within about a 3-month window. And so this allows us to, in the summertime, determine, what viruses are circulating, what's the variant of COVID-19 that we need to target, create a vaccine, and have that available so we have a nice, closely matching vaccine, as opposed to a big mismatch like you sometimes have with the influenza vaccines. And this is something that, mRNA technology is being looked at for influenza as well for this exact same reason. It's trying to see, is there a way to have more effective influenza vaccines that are able to target more closely the circulating variants. The other thing is, customization is that you can really target sometimes difficult or even previously untreatable diseases. I mentioned cancer before. You're able to actually target that person's own cells that are expressing kind of these strange proteins that are only being expressed in that particular cell, and allows you to specifically target. And that's something when you use a typical immunotherapy, it's really hard to target the cells only that have cancer, and sometimes have a lot of off-target effects and a lot of side effects because it's not just killing the cells that are cancerous. And then again, the platform technology, again, the fact that we can use the exact same ingredients, manufacturing equipment, everything to create multiple different vaccines. And so when you look at the other mRNA vaccine that's approved for RSV, the only difference between the RSV vaccine and the COVID-19 vaccine is just the sequence of the protein that's targeted. So instead of targeting the spike protein, you're targeting from RSV the prefusion F protein. And so, but everything else, the lipid nanoparticle, everything else that's kind of contained within that is pretty much the same. And those can both be manufactured with the same ingredients and the same equipment, of course, at different times, but you get that flexibility there. And so those are kind of the main advantages of using the mRNA technology. And I think as we gain more and more understanding of it, I think we'll identify even more advantages, particularly for certain therapeutic areas where there aren't currently any treatments available.
I'd like to flip the conversation just a little bit, Kyle. Of course, with the pandemic and public perception of hearing about mRNA vaccines for the first time for many people, there was some hesitancy around, I guess, trust in it and the development of mRNA vaccines being too rapid. So I'd like you to comment on that. And as well, are there any other downsides to using mRNA technology?
For sure. And as we kind of talked about before, development during the pandemic was very quick. And part of that was really because a lot of streamlining of processes that normally take a really long period of time. And so we were able to really fast track, getting a clinical trial on track and getting patients enrolled in that. We're able to work with the regulatories to review simultaneously the efficacy and the safety data that was being generated for approval. And so really there weren't any steps that were skipped. It was just we kind of had a very efficient process throughout the pandemic where the main goals of everyone is really to try to identify and create a vaccine that was effective as quickly as possible. But going back to what we talked about at the beginning, this technology has been researched for decades prior to the pandemic. And so Moderna itself actually was created back in 2010. So Moderna had been working on mRNA vaccines for a decade prior to the actual pandemic starting. And really, it started off with looking at mRNA vaccines for pandemic influenza viruses, because at the time that was kind of our biggest concern. Looking at it in other kind of viruses and therapeutic areas. It's where the kind of the research in rare disease and things started. And it was really just the timing that when the pandemic occurred, the technology had actually matured enough to actually be able to be used in this type of context. And that if the pandemic had happened, 10, 15 years earlier, the technology wouldn't have been advanced as much to be able to actually be used in this situation. But there are some downsides and limitations of mRNA technology. And so one of the biggest ones is that mRNA is rapidly degraded. And so we talked about before why that's an ideal thing in the body. We don't want it to be sticking around for a long period of time. But that's not as ideal when it's outside of the body and it's in a vial. And so this means we have a number of stability challenges to manage. A lot of times our vaccines do require ultra cold storage, so specialized freezer units that can keep it cold for long periods of time. But there's been a number of advancements that are currently in trial and being reviewed that allow these products to be more stable at refrigerated temperatures. And that helps with a lot of the cold chain logistics. The other thing is really, we've touched on this as well, the public misunderstanding that they're really only hearing about mRNA technology over the last couple of years here. And so there's a lot of misinformation and hesitancy. And a lot of that is because even healthcare providers don't have a great understanding of the technology. And now that we're out of the pandemic, we're hoping that'll change. We're hoping that'll change as more vaccines and therapies using mRNA come out. But again, there's going to be that initial apprehension, about any kind of new medication or technology that comes out. And finally, the main thing is it's not a mature technology. We know that it works very effectively for prevention of COVID-19, but there's lots of things that can be improved for future therapeutic and vaccine areas. So currently, there's being research on making changes to the lipid nanoparticles, making changes to the coding sequences to make them more effective, to more specifically target, certain cell types, and maybe alternate routes of administration as well. So not just intramuscular injection, but through, other administration routes as well. And there's also research being done on self-amplifying mRNAs. And so these are mRNAs that they themselves can amplify in the cells and not just amplification of the protein. So there's lots of things that will create version 2.0 and version 3.0 of the vaccine platform. The advantages of you get from some of those conventional vaccines that have been around for decades, these aren't the first iteration of those vaccines. That's a very mature technology. And so as much as there are certain limitations with the technology now, that will improve over time as more and more research is done in how to make these changes to the technology.
Well, Kyle, we know that today mRNA vaccines are used to prevent COVID-19, with NACI recommending COVID-19 vaccines for at-risk patient groups each fall. Tell us why it's important that people need to continue to get COVID-19 vaccines each year.
Sure. So, like the flu, which we've talked about, the virus evolves on an annual basis. There's constantly mutations that occur because the virus wants to evade our immune system. It wants to prevent getting destroyed and actually be able to cause an infection. And so by making these changes, it's able to better infect us. And so in order to be prepared to protect ourselves from it, we need to also update the vaccines so that these vaccines better match the circulating strains. And this is also because immunity wanes over time. You have a decrease, especially in the older adult population or people with other comorbidities. They may not have as strong of an initial immune response to the actual vaccines. And so, data that we have from both Canada as well as the US shows that receiving an updated annual vaccine is highly effective at reducing the risk of severe outcomes like hospitalization and death, even compared to individuals that have been previously vaccinated or infected but did not receive the updated dose. And so the protection that we're seeing isn't like at the beginning of the pandemic where you're comparing to people that got the vaccine versus that didn't get the vaccine. You're actually saying that even people that have had all of the previous doses up until the current year, there's still an advantage for those individuals to get the updated dose compared to those that elect to not get the updated dose that year. And so NACI continues to recommend fall vaccinations to reduce severe outcomes, particularly in the vulnerable populations. The groups, again, that are 65 plus, that are in congregate living settings like care homes, for people that are pregnant, and for people that have other comorbidities, so diabetes and lung disease and heart disease and kidney disease and all of those other things. It's important that they are targeted to get the updated vaccine on an annual basis. And finally, unfortunately, like flu, COVID has become endemic, which means it's here for the long term. And so just like with influenza, high-risk populations do need to receive that updated dose on an annual basis and biannual in the case of COVID for certain individuals. And it's important to note that this isn't a limitation of the mRNA vaccines themselves, but is more of the rapidly changing viruses. And so future mRNA vaccines against more stable viruses may require less frequent dosing. Again, as an example, the mRNA-based RSV vaccine is currently only approved as a single dose. And And a lot of research is being done with the mRNA RSV vaccine as well as the protein-based RSV vaccines to determine when an additional dose of the vaccine would actually be required. And so this is evolving, but it's important to note that this is the reason why you have to get these doses on an annual basis, is it's because of the actual changes in the viruses each year. And as the viruses change, maybe the recommendations would change as well, but that's still something that we're watching on a year-by-year basis.
Kyle, we're winding down our time together with you today, but we just have a couple of more questions to ask you. Tell us about what is on the horizon in mRNA technology. You did mention themes such as personalized precision medicine. We're just wondering where else we might be seeing it used and how is it being researched right now?
Course, there's a number of different ways that it's being looked at. There's additional. I've mentioned some of the respiratory vaccine targets already with RSV, which is currently approved, but there's also influenza vaccines that are being evaluated with mRNA technology. One of the cool things is looking at combination vaccines. So instead of getting a single vaccine at a time or multiple vaccine shots at the same time, you can actually get a single dose or a single vaccine shot that includes protection against multiple viruses. And so that makes it more convenient, especially because a lot of these respiratory viruses have the same kind of high-risk patients that need to be protected. But there's also being, research done with non-respiratory viruses as well as bacterial infections. Some of the non-respiratory viruses include like cytomegalovirus or CMV as well as norovirus. There's the cancer vaccines, which I mentioned as well. So that's really the personalized approach to stimulate the immune system against that particular person's cancer cells. And this isn't just usually mRNA on its own. It's been looked at as mRNA with the conventional immunotherapies. And so it's kind of an added on benefit in terms of the current standard of care for the treatment of these patients. And then rare genetic disease, again, is a really great area to be looking at, because for a lot of these individuals, they're not able to effectively metabolize a specific product that they take into their bodies, a lot of times through what they're eating. And so those molecules can kind of build up to toxic levels, and they can have really bad seizures. They could potentially even die from it. And so by using mRNA to replace temporarily those, missing or faulty proteins, it can actually make a huge difference in that individual's life, that they can now metabolize those products, and they can prevent, seizures and other kind of adverse outcomes. And it also gives us a lot of information in terms of repeated dosing, because those individuals, unlike vaccine individuals where you get maybe one dose per year, these people with rare diseases are provided the mRNA very frequently. And so it kind of gives us an idea about the safety of increased dosing of the technology as well.
One final thought from you, Kyle. We're just wondering, how is Canada showing leadership in the advancement of mRNA technology?
One of the exciting things is that, Canada is really establishing itself as a leader in mRNA technology. And that includes a number of different factors. One of the main things for Moderna specifically is we actually have a manufacturing built facility. We have a manufacturing facility that was built in Laval in Quebec. And so the actual mRNA vaccines are created there and then moved to Ontario with our fill finish partner there to actually create the vaccines themselves. And so Canada will actually now completely possess an mRNA vaccine production supply chain, which is fantastic for, future pandemic preparedness. There's also a lot of different initiatives across the country. Quebec has their ARENA initiative, which is the Alliance for RNA Research and Innovation. This is really looking at advancing RNA science and accelerating research in this area. McGill University was given a grant from the federal government really to establish the McGill Advanced RNA Sciences Initiative, which is expanding Canada's leadership in RNA discovery and manufacturing and commercialization. RNA Canada is a national nonprofit that's also working with academic and industry labs from right across the country. And there's also partnerships with McGill, University of Toronto, UBC, researchers right across the country that are working on this technology. And finally, clinical trials as well. There's a lot of clinical trials for not just Moderna's products, but for other companies as well that are happening in this country to really advance the use of mRNA-based medicines.
Kyle, talking about science and explaining science isn't always easy. You have taken quite a difficult technical topic and made it so meaningful for us. We can't thank you enough for joining us for today's episode. We sincerely appreciate you, the important work that you're leading for those living with lung disease or facing threats to their lung health, and want to sincerely thank you for being with us on this episode of Let's Talk Lungs.
Great. Thanks, Trent, and thanks, Lori, for the opportunity to have a chat here today.
Thanks for listening. If you enjoyed this episode and you'd like to help support LungSask's Let's Talk Lungs podcast, please share it with others. Post about it on social media. Like, subscribe, hit that notification bell for new episodes, and leave us a review. Follow LungSask on Instagram @LungSask and on X @LungSask. That's all for this episode. See you next time.
Produced at
Sound Lounge by T-Bone.